Hello everyone! Today, we're diving into a fundamental technique in biochemistry called dialysis. Have you ever wondered how scientists clean up a protein sample, removing tiny, unwanted molecules like salt? The answer is often dialysis, a simple yet powerful separation method. At its core, dialysis works on the principle of selective diffusion across a semi-permeable membrane. Imagine a special bag with microscopic holes. These holes are just the right size to let small molecules pass through but are too small for large molecules like proteins or DNA to escape. This size-based separation is the key to the entire process. So, how does it work in the lab? First, you take your sample, which is a mix of your large target molecules and small contaminant molecules, and place it inside a special tube or bag made of this semi-permeable membrane. This is called the dialysis bag. Next, you submerge this sealed bag into a large container filled with a liquid called the dialysate, which is usually a pure buffer solution. Once the bag is in the dialysate, the magic of diffusion begins. The small, unwanted molecules, like salt ions, are highly concentrated inside the bag but have a very low concentration in the surrounding dialysate. Following the natural law of moving from high to low concentration, these small molecules start to diffuse out of the bag, through the tiny pores, and into the larger volume of liquid. The large protein molecules, however, are trapped inside because they are too big for the pores. To make the process really effective, the outer liquid is often stirred and changed a few times. This keeps the concentration of small molecules outside the bag very low, encouraging more of them to leave the sample. After a few hours, or perhaps overnight, you are left with a purified sample inside the bag. The large molecules are still there, but they are now in a new, clean buffer solution, free from the small contaminants. This is incredibly useful for preparing proteins for experiments, a process known as buffer exchange or desalting. It ensures that the conditions are perfect for studying the protein's function without interference. And that's the essence of dialysis in a biochemistry lab. A straightforward but vital tool for purifying macromolecules. Thank you so much for watching. If you found this explanation helpful, please consider liking this video and subscribing to support the channel. It helps us create more content just like this. See you in the next video.